Hydrolysis of C3 causes initiation of the alternative pathway of complement. The third pathway of complement activation is called the alternative pathway because it was discovered as a second or alternative pathway for complement activation after the classical pathway had been defined. This pathway can proceed on many microbial surfaces in the absence of a specific antibody, and it leads to the generation of a distinct C3 convertase designated C3b-BB. In contrast to the classical and MB-lectin pathways of complement activation, the alternative pathway does not depend on a pathogen-binding protein for its initiation. Instead, it is initiated through the spontaneous hydrolysis of C3. A number of mechanisms ensure that the activation pathway will only proceed on the surface of a pathogen. C3 is abundant in plasma, and C3b is produced at a significant rate by spontaneous cleavage, also known as tickover. This occurs through the spontaneous hydrolysis of the thioester bond in C3 to form C3H2O, which has an altered conformation, allowing binding of the plasma protein factor B. The binding of B to C3H2O then allows a plasma protease called factor D to cleave factor B to BA and BB, the latter remaining associated with C3H2O to form C3H2O-BB complex. This complex is a fluid phase C3 convertase, and although it is only formed in small amounts, it can cleave many molecules of C3 to C3A and C3B. Much of this C3b is inactivated by hydrolysis, but some attaches covalently through its reactive thioester group to the surfaces of host cells or to pathogens. C3b, bound in this way, is able to bind factor B, allowing its cleavage by factor D to yield the small fragment BA and the active protease BB. This results in the formation of the alternative pathway C3 convertase, C3b-BB. When C3b binds to host cells, a number of complement regulatory proteins present in the plasma and on host cell membranes combine to prevent complement activation from proceeding. These proteins interact with C3b and either prevent the convertase from forming or promote its rapid dissociation. Thus, the complement receptor, CR1, and a membrane-attached protein known as decay accelerating factor, DAF, compete with factor B for binding to C3b on the cell's surface and can displace BB from a convertase that has already formed. Convertase formation can also be prevented by cleaving C3b to its inactive derivative IC3b. This is achieved by a plasma protease factor I in conjunction with C3b binding proteins that can act as cofactors such as CR1 and membrane cofactor of proteolysis MCP or CD46, another host cell membrane protein. Factor H is another complement regulatory protein in plasma that binds C3b, and, like CR1, it is able to compete with factor B and displace BB from the convertase in addition to acting as a cofactor for factor 1. Factor H binds preferentially to C3b bound to vertebrate cells as it has an affinity for the sialic acid residues present on these cells. By contrast, because pathogen surfaces lack these regulatory proteins and sialic acid residues, the C3b-BB convertase can form and persist. Indeed, this process may be favored by a positive regulatory factor known as properdin or factor P, which binds to many microbial surfaces and stabilizes the convertase. Deficiencies in factor P are associated with a heightened susceptibility to infection with Neisseria species. Once formed, the C3b-BB convertase rapidly cleaves yet more C3 to C3b, which can bind to pathogen and either act as an opsonin or reinitiate the pathway to form another molecule of C3b-BB convertase. Thus, the alternative pathway activates through an amplification loop that can proceed on the surface of a pathogen, but not on a host cell. This same amplification loop enables the alternative pathway to contribute to complement activation initially triggered through the classical or MB-lectin pathways. And that's hydrolysis of C3 causes initiation of the alternative pathway of complement. Have a great day.